Welcome to Channel 7 Action News. Did you know that over 75% of all adults don't even know about protein synthesis? First off, DNA molecules are polymers. Polymers are large molecules that are built up by repeatedly linking together smaller molecules called monomers. DNA monomers are called nucleotides. There are 4 monomers: adenine, cytosine, thymine, and guanine. DNA structure can be described as a double helix, antiparallel, 3D shape. The helix of DNA is connected by hydrogen bonds. Since DNA is antiparallel, there's a 3' end and a 5' end. And on the other end, there's a 5' and a 3' end. DNA's function is that it contains genetic instructions for the development or function of living things.
Fun.
Here are some images of DNA structure and what they look like. So these are the 4 bases: adenine, thymine, guanine, and cytosine. As you can see, they are connected by the hydrogen bonds within the double helix structure of the DNA. There's a 3' and a 5' and a 3' and a 5' end here. There's sugar phosphate, backbone, and a base pair.
Hi, I'm Joe Cable from Channel 7 Action News. Today we'll be talking about DNA replication. DNA replication is where DNA copies itself into end-cell division. DNA replication starts with the unzipping the double helix. DNA helicase is responsible for unzipping it. Next, the separation of 2 single strands of DNA creates something called a replication fork. The strands are used for making new strands of DNA. One of the strands is the leading strand, which is in a 3 to 5 direction. The other strand is in a lagging strand, which is 5 to 3. Since the strands are different, they are replicated differently. In the leading strand, the primer binds to the end of the leading strand. The primer is the starting point for DNA synthesis. DNA polymerase binds the leading strand, then adds new complementary nucleotides in a 5 to 3 direction. In the lagging strand, RNA primers are made by primase and bind to points on the lagging strand. The strand is proofread so the DNA sequence has no mistakes. Finally, ligase seals the sequence of DNA up in a double strand, and that's how DNA replication works. These are the pictures that represent DNA replication.
Transcription is when the DNA in a gene is copied to make an RNA transcript called mRNA. The process is carried out by an enzyme called RNA polymerase, which uses the available bases from the nucleus. The 4 bases that can be used in this process are adenine, cytosine, guanine, and uracil. Translation occurs after the messenger RNA has carried the transcripted DNA or message from the DNA to the ribosomes. The message carried by the mRNA is read by a carrier molecule called transfer RNA or tRNA. Each codon specifies for a different amino acid. For example, the 3 bases AAG codes for the amino acid lysine. When the mRNA sequence is read, each tRNA molecule temporarily binds to the corresponding codon on the mRNA. The tRNA is bound, it lets go of its amino acid, and the adjacent amino acids all join together into a long chain called polypeptide. This continues until a protein is formed.
Lastly, I would like to introduce you to our sponsor, DNA Legends, the most ambitious mobile game available in the Apple Store and Google Play. New updates daily. Use our promo code Channel7News for a free DNA strand. New factions mode coming out soon. Download it now.